Welcome to today's lecture. As I said last time, the last lecture will be July 7th, examine July 9th, most likely in this room, makeup October 13th. Those are the holidays. Last week we discussed 3D comparisons and 1D comparisons. In particular, we began with simple alignments, pairwise string alignments. Today we will get into multiple alignments. Recap, science is communication. Please interrupt and ask questions. On the notion of 1D, 2D, 3D, the structure, protein structure, are the 3D coordinates. That's typically what we refer to as the protein structure. So where every unit of that protein residue, amino acid, has a position in a 3D space, a coordinate. That's 3D. 2D, simply for all these residues, for all these units, you look at the distance of all the others. In this particular case, the protein is 98 residues long, so at every single point you look at the distance of all the others. You see that most of the residues are close to the diagonal, so they are contacting only their neighbors. I said that the 2D representation fully contains the 3D information. You can essentially reconstruct the 3D structure from the 2D image, provided it's complete. Further reduction 1D, just onto strings. That loses the important information about 3D structure. It still maintains some aspects that we will get back to later, such as secondary structure and where in the protein you are solvent accessibility. Protein structures, there's not one solution. For one particular protein there is a solution, but there's dynamics. Some regions of this protein move more. What you see here is a superposition of an entire family of 300 structures. And although it may look messy to you, if you remember, sort of these red pinkish things are helices, blue are the strands, and you see that although you have 300 superposed here, essentially the strands at least fall on the same place. And even the helices, you still can sort of recognize the winding of a helix. Winding of a helix means they're not even one residue off. Otherwise, you would just see this as a long rod. But you can really see winds. Okay, you have many different traces here, and they have regions where the whole thing looks like a wool ball, ball of wool, or hair ball. But still, there's a lot of similarity. The simplest way to align strings is brute force. You simply try all the possible ways in which you can superpose these two strings, and the matrix gives you a simple way to do that. That leads to dynamic programming. Dynamic programming is fairly fast in the square of to the two length of the two sequences, a very fast algorithm to align sequences. But much faster if we think about aligning databases of 50 million sequences, one against each other, so 50 million times 50 million, then you need to speed up much beyond this dynamic programming. And that is what you do simply through hashing. So you hash words. In this particular case here, these words have a length of 3. Every possible word, you look for an identical word match, and since that is a hash, it's very fast. And then you extend from these initial hits of identical letters, 3 mers, into both directions by dynamic programming. This is much faster because now you extend on a scale of, say this is 10 residues, 10 by 10, rather than the whole thing here is 100 by 100. So you gain immense time by cutting the problem up. The problem that you buy yourself into is that if you look at a database of 50 million sequences that are about 400 residues or something like that long, then any letter may hit at random. So you have to have a good model for what is the background. That is the background distribution. You have to have a good way to model the background distribution. You cannot, for every single alignment, compute the background distribution, because then you have to again do the same alignment algorithm. So you have to find a good way to approximate it. And then the distance of the hits from the background distribution tells you how significant that hit is. I told you that there is a zoning in the comparison of proteins. So there's a zone, the safe zone up here, where comparisons are very, very simple. Typically in terms of sequence identity, so that's percentage of residues that are identical, or amino acids, from 100 to around about 40, the daylight zone is where everything is sort of easy. Everything easy means you can recognize the two proteins that you align have a similar structure. That, for the course of this lecture here, is the main goal of that method. And in that sense, it's a daylight zone. In that sense, it's simple. Then we get into the twilight zone. In the twilight zone, you begin to lose the signal. You still have a lot of findings here, but many things that you have now are wrong. So you work in your signal against the noise, and in the midnight zone, totally dominated by noise. Okay, so far we aligned pairs. In this image of a family I already showed you, in 3D, this is a solution where you superpose many structures in 3D. In order to superpose many structures in 3D, you essentially do what I showed to you a week ago, which is you do pairwise superpositions and you do it again and again and again. There's no problem to that. Now, for the alignment of sequences, the story is slightly more complicated. Because now you don't necessarily want to find, so you line one against the other, and now you want to bring a third sequence in there. But the alignment should be optimal for all three of them. Okay? In order to do that, you essentially would have to take this matrix here and expand it into a 3-dimension. So you have essentially a 3D matrix. And that begins to be complicated. One immediate problem here that you see here is that it's in the three lengths, n1, n2, n3. So previously we had only n square, and now we have n cube to cope with. And that is a problem if you have proteins that are typically, but sometimes 1,000 residues long. It is, in fact, an NP-complete problem and requires a lot of memory. So we need a hack. And so I'm showing you that the problem is very difficult for 3. On the slide before, I showed you a family of 100. So the problem is already difficult for 3, forget about 100. What I want to do is I want to align 100 proteins. I know how to align pairs. How could I come up with an algorithm with a hack that does the family? Any idea? Well, the simplest solution is a stepwise process in which you begin with 2. These 2 you align according to what you did before. Then you put another one in there and align it to those 2. And now when the third one here, the green one, comes, you have 2 choices. One is you could align it to what is typically referred to as the consensus. So you write down a sequence here that is the majority of letter that you encountered. So here in this particular column I just chose the E because that is the first. If we had 3, you will see then one of these 2 may be the majority letter, and then you write that. And you simply take this pseudo sequence. In this particular chosen example, this pseudo sequence is the actual sequence of the first protein, but that does not need to be the case, in particular not if we go further, right? And if these sequences here really differ. My toy example is a little bit too simplistic in this. But anyway, so you could imagine that what you take now is this consensus sequence here, and you line the third one into that consensus sequence. So you line it neither against this nor against this, but against some average between the 2. As simple as that. Then we are back to this problem that we already know. Pairwise alignment. Same letter, same alphabet, everything the same. And again, in this particular case here, you again compile the consensus, which in this particular case, again I should change my example here, looks the same. And you come with a fourth and you line it into that consensus. And you keep going. Once you have built up one round, you write this matrix here. The matrix essentially shows what's the overall similarity or identity in this particular case between two proteins, between two strings. A and B, in this example you have 90% of their letters identical. You need to do that because now you need another round. In the first round you assume that this is sort of the start and everything is aligned to that, and then you somehow go randomly down the hill. So you pick the second, the third, the fourth. In the second round, you should have some more rational picking. And that more rational picking could be simply you go by the highest number. So in this particular example here, you could first align A and B, or A, B and C, or C and D. Let's pick this one. Now once we have that aligned, we need to find the next best. The next best would be B and C. Or A and C. Again, it depends on how you continue. But that gives you the order of the matter. And then you build up again. The second way of doing it may lead to differences. In this toy example, it will not. Because in this toy example, all the sequences have similar length. They have all similar residues. They're very, very similar. But the moment you include more changes in there, most likely there's something going to change. So this gets into the next idea, and that is the idea of profiles. The concept of profiles really is you build up, what I showed you was a consensus sequence. Now you could also imagine that you essentially build up something else that describes these two sequences here, and align into that something else. That something else, when you look at multiple alignments, I showed you images like this where the protein runs from here to here. One line means the beginning and end of a protein. Overlapping lines, this one here, the third and the first, means over the region where they overlap, they have essentially similar structure, or the sequences are aligned and align above some threshold. That's what the lingo means. And I said before, this sort of is indicative, if you see an alignment like that, it's indicative of a domain here and a domain here. Now, what it tells you already is that what you see there has different. Is that a question? That's very subtle.
Yeah, sorry, just a couple of slides ago you said average between the two sequences. How do you get that?
Well, so average is the wrong word. There are different ways of doing that. So the simplest way, and that's the one for my toy example, I will present other examples to you later. The simplest way is that you look for every position, what is the majority that you find? In most of these cases, trivial, because of most of those cases, both of them have identical letters. Where they differ, E and D here, the story begins to be more complicated. Now you have a choice. You can either, in this particular case, take E or D. That decision could be driven by what is first, because that is sort of the query sequence with which you start. It could be driven by, say, there would be more than two. So consensus sequence comes also from more than two, then the majority. Or it could be driven by something totally different. You could argue that at this particular position, you have a random probability of a particular residue over the course of an entire group of sequences. And random essentially now means the amino acids occur at a different frequency. And now again, you have two choices. You could look at the frequency as the entire protein. So what's the probability of getting an A in that given sequence of 1,000 residues? Another way of looking at it, what's the probability of getting an A, say the query is a human protein, in all human proteins? The third way, what's the probability of getting an A in all proteins? That probability you write down. And then you essentially do not take the one that has the highest count, but the one that is most surprising. Has the occurrence of which has the highest information content. That's the least likely to happen by chance. Okay? And I believe I went into so much detail in answering your question in order to illustrate something. It's a very simple problem, and I just used a word averaging over letters, which you cannot do. And the concept is simple. and you immediately are faced with so many options, and it's absolutely not clear a priori which one is best. Unfortunately, in computational biology, often it also doesn't work that you say, well, let's just try them all out and then see which one works best. Because works best depends on many, many other aspects. For instance, what particular family you have in front of you. Okay, let's get back into the idea. So you see that we have some, let's misuse the word structure here, some grammar or whatever you call it. I'm missing the right word here. Structure would be the right word, but in the context of protein structure, I don't want to take it. What I mean is you have some region here where you have density, and some region where you have density, and some region where you have lower density. That's simple. That alone, just with the black lines, contains information. From that alone, you see that the probability of now, when you come in with a new sequence, aligning it here, in principle, should be higher. If you don't know anything about the sequence that comes in, and if you have not even looked at letter comparison, the simplest way you can do is you mirror the density distribution. That's the best random background model that you can have, given this observation, right? That means that in this alignment that you have, there is more information contained than just the consensus sequence. Let me give you an example, and that slide comes from Teresa Wirth. Here is a transcription factor binding site. Transcription factors, they are the ones who decide how genes are transcribed on the genome. So that's why you see essentially only 4 letters. The gene, the nucleotides. On the genome, the transcription starts at position 0, and downstream, upstream, you have a probability of observing certain nucleotides. The height of the letter is proportional to the probability of observing that letter at that position. And you see immediately, that's a profile. You immediately see that T and G are highly abundant, more than background probability at -6, -5, -7. Here's a cluster, and here's a cluster, CAG. So that's highly informative if you want to bind to that transcription factor binding site. Then that is the motif, or if you want to find a new binding site, such as this one, that's the motif you're going to look for. Motif, simply, you put in some model the probability of these observations here. Okay? And that's a profile. Now, here is another way of formulating a motif. You could also formulate it, just that is the sequence, by saying, well, M, the methionine is one letter. In this particular position here, you have 1 or 3. So it's a regular expression motif, right? Here you have 2 or more possible. Here the L or I. X, this is applying here. The disallowed, the X means, I'm sorry, any means, my arrows are off. They were right. That's one of those things in life. And that's the repetition here. So you essentially have a simple regular expression. Different sequences can map that regular expression. Yes? Colin.
In the previous slide, you explained the axis.
This one. The axis simply is 0 is where the transcription factor binds, and minus is the nucleotides away from it. So that is the transcription factor goes to the DNA. The DNA is rolled out from, in this particular case, what you see is -9 to +10, so 90 nucleotides. And what you're asking, what this essentially answers, is the question, when you bind as a protein to that piece of gene, where are you going to bind? That is the binding site, but the binding site itself is not really characterized by there's no clear binding motif in the binding site. The motif actually comes from left and right, or downstream and upstream and downstream. That direction of the reading. Yeah? Thank you. So back again, simple regular expression. The regular expression also defines motifs, but the difference between this and the letters that I showed you before, this is also a position. Now we have you could do both on proteins and nucleotides. Here I have the protein example, previously I had a nucleotide example. But the difference here is now typically you're much more confined with what you have at which position. And you don't have a probability model. This is either or. There's simple rules. Here is another example of a profile. Let's call it generalized profile. That's the substitution matrix. I told you that you can align a bunch of proteins, and then once you have them aligned and you know that they are similar, you can simply count what is the probability of exchanging an S and against a T, and so forth and so on. You compile log odds and you come up with a matrix that overall gives you the probability of the information content of particular amino acids exchanged to other amino acids. What is observed in nature? This one reflects. This is also a profile, but it's a very, very generic profile. It's the best thing you can do if you don't know anything. So you match two amino acids, two letters, and matching one letter onto the other may not be the best option. The best option may be to look up in this substitution matrix what is the observed probability in nature to see the substitution L to I. Again, this is not a profile that is specific to that alignment that I showed you before, but it's a generic profile. In order to get a specific profile, a position-specific substitution matrix, you simply begin with, and this is again our transcription factor example here, you align a bunch of nucleotides, then you compile the frequency, you compile the relative frequency that gives you a matrix at every single position, 1, 2, 3, 4, 6 here, for each one of these 4 nucleotides, observed probability, and that probability with log odds you can compile into a matrix that is like the one before, but that one is specific to the example that I showed. That now is specific for the set of sequences that I found. That is specific to one particular protein family. The way then it would read in a protein example, so you would run typically here the number of the residue. That's very similar to the slide 1D, 3D, 2D, 3D that I showed you. And then you have an alignment here. An alignment means that is protein 1, 2, 3, 4, and they all run down here. And at position 1 you have E, G, V, L. So we have 4 different proteins aligned. One can be E, 1 G, 1 V, 1 L. There's a consensus, and now you already see an example where the consensus is more clear here. VLVA, the consensus sequence here would be a V. And you can compile from that, just like in the example that I showed you before, a log odds matrix, you can compile a profile. Now that's a position-specific profile. That's valid for that particular alignment. Comes from that alignment for that alignment. And that essentially is what is behind a program called PsyBlast, the position-specific iterative basic local alignment tool. And essentially it does exactly that. It finds regions. In purple you see here highlighted 4, it's hardly visible, but essentially this is a very diverse alignment, and 1, 2, 3, 4 columns are fairly conserved. When you come up with a new sequence, you will try to have it match such that these things will maintain their conservation. That's the most important thing in the alignment. So you will go from the generic scoring matrix, the BLOSUM, to the specific matrix that you look up from that alignment. The concept of PsyBlast is the same as before. For BLAST, you have fast hashing. So first, you find short, identical matches. No profile involved at this point. Just look for letters. Simple hashes. You expand as before, like with dynamic programming, in both directions. You apply statistics, but now you collect all the pairs and build up the profile. That should be this one here. Compile a position-specific matrix, and now with the new sequence, you align into that matrix. Actually, the first thing would be to simply redo this profile, as I showed you before. But now we come with we we have a specific matrix that is specific to that family. So now what we can do is we can look further away. That is the story about intruding into the twilight zone. With pairwise alignments, we can just go in front of the twilight zone without making mistakes. Now we have a specific matrix. That means we can go a little bit further down into the twilight zone. We can find things that previously we were not sure would be right matches. The profile allows us to go and take them. So we can go further down, collect new matches, build up a new profile, and iterate. Redo that. So every single time we go a little bit further out, we find that, change our profile, that allows us again to go further out, and so forth, and so on. Now, there is one problem that comes with it. When you expand the sequence space in this way, the way you can think about it is you start with the protein U. Say this green cloud here were my protein family. Everything on that green cloud in principle would be something that I would like to reach by my alignment method. In my first step in pairwise alignment, essentially what I do is I draw a circle around U, because I just look for pairs and I can see everything that is within that circle. There is no structure. Assume that here's my U, that's my U is at the center, sorry. In my first step, I find those 5 first hits. And they happen to be on that side of the circle. This may simply be because that's the way they cluster. They are maybe more variants of the first part of my sequence than of the second. And all the guys who vary the second part would lie here, but I don't find them in today's database. At least not with the search I've done so far. That's a simple pairwise alignment. What that leads to is a profile that zooms into this area here. It will be wider than the area of the first circle. The profile allows me to go further without making mistakes. It will be wider and it will be moved away slightly from the center. U will no longer be at the center of this new lens because it is biased by what it found. And what it found in this particular example is on the right side. In my next, that in particular implies that if I had found here one similarity in the safe zone, or if tomorrow I started with that profile and there would be a protein in here, then I would no longer find that. The profile does better, looks wider, more specific. Still it misses things. What initially was safe is suddenly away, simply by the fact that this is sort of biased in that direction. So now we go further, and by the way, another thing that we are missing in this lens here is if I had started the search from every one of these white dots, and every one of them would be a circle pairwise comparison around these white dots, you see that these white dots here, or these light blue disks, expand beyond that lens, and that could really be the case, but it is also much slower. 5 searches instead of 1. But now assume I make my lens even bigger. Assume I find one protein that comes from a different family. So that's an incorrect hit. I made a mistake. I made my lens a little bit too big. And that now is the pollution that comes immediately. The fact is, as long as I stay on this island, on this family, whatever I find is specific to that family. I showed you this example here where we have 4 entirely conserved residues. If I believe a protein is similar to that one here and that does not have any of these 4 conserved, then in my next step the profile will be totally different. Because it has violated what has been essential building up this family so far. In this particular case here, you see, I believe, 30 or 50. This is from an alignment of hundreds of proteins. One sequence can completely poison that. And that is why when you put in the wrong one here from a different family, the profile completely explodes. The problem is you will not be able to anticipate that. You will not see that. Unless you know something about the proteins that you don't know, because then you wouldn't have to align. You could argue, well, maybe monitor that at a particular point of the iteration I get so many new ones in. The problem is it could also be that simply your lens gets bigger, and that's why you get so many new right ones in. Because you explore parts of the island that you previously couldn't reach. That could also happen. So one giveaway would be if you suddenly align the whole database. That's clear. Or you could imagine other hacks. You could put in random sequences, and you shouldn't find those. But now we get into the world of hacks. But this is one problem with this iteration. Clustal W, Clustal X, Clustal Omega is the type of program that now does something like PsyBlast, so it uses profile-specific, position-specific matrices in a dynamic programming. The advantage is it does it well. The disadvantage is it's very slow. By the way, Clustal historically was there before PsyBlast, and Clustal is in fact one of those methods that invented this idea of position-specific profiles. There's a generation of Clustal. The latest one sort of started in '88. The latest one is Clustal Omega, and that comes as, with the description, is the last, oh, I don't have the quote here, is the last alignment program you will ever need. What Clustal does is essentially implements the hack that I showed you before. It does an all against all by some simple algorithm that could in fact even be a blast. It doesn't do that, but in principle it could be a blast, something fast, just to get here these numbers. Once it has these numbers, so what is the most similar one? Let's begin with the most similar one. It builds a tree. In the tree, it begins with the most similar pair, and then the next most similar pair that is disjunct, and builds up a tree from there. Now this brings me back to phylogeny. The idea of phylogeny and what we do with it and what we can do. In principle, evolution is this way. Again, by now you understand my graphs. So the blue line and this blue line essentially means there's something repeated. In this particular case, what I mean here is that I have a protein or a gene, and then I have some duplication event. What happens frequently is that in fact entire regions of the genome are duplicated. You suddenly have the same thing again. That could happen under stress. So if you put yeast under heat stress, a large chunk with 100 genes is immediately repeated. Yeast, by the way, has about 6,000 genes, so 200, quite a substantial portion is duplicated. And this could happen. Now, in the next step of the next organism, we reproduce, we copy the whole thing because we had it, and we see that actually my first region here, and the next one we realize that the first one here is containing a domain, and that what you actually did is you duplicated the domain. Since you duplicated the domain, the light blue here indicates the original domain is there, and something more. Since you have something more, you can begin to deviate and to slightly change that. The original function is done by the light blue. So the organism has the freedom to evolve a new function onto this dark blue here. Now say this again, now you have a two-domain protein, and that would now, or two-domain gene, that would be duplicated. So 4. Again, have the light blue even twice, you have this entire region that you can change, and let's say at some point here that would be changed in this red region. Another story where we in fact begin with a green, this long green chunk. We see that has a duplication of 2 domains. Only one is needed, or only one is essential. The one is a little bit elongated, a new domain comes, a duplication event comes, things move around, just in the same story as before. At some point, this is what you see. Story 3. You have this pair, and you put it together in 2 different ways. Since it's put together in 2 different ways, you may not need that, and you in fact may evolve to this construct. Our view of these stories may be just that. Maybe all we see today in the sequences, maybe all these things that I showed you that evolved no longer exist except for that. If you saw this, what you would be inclined to do is simply align them this way. And conclude, according to these colors here, what is evolutionarily related. And that would be, according to my example that I constructed here, absolutely wrong. This is similar in sequence, and from the similar in sequence I infer similarity in evolution. But I did that because I did not know the story, because I lost most sequences. Most creatures that ever have lived died. Most of the creatures that live today have not been sequenced. When we scoop up a piece of dirt, we find new sequences in an amazing amount. Most of the organisms that live in the soil here are not characterized. Most of the sequences that exist around us on the planet are not known. Most of these trees, most of these stories are not known to us. Nevertheless, we try, from what we observe as sequences, to reconstruct evolution. And that is a daring enterprise. Surprisingly often we get somewhere that is remarkably close to what most likely was truth. But it's a very, very difficult one, and we have to remember how much of a fiction we are constructing here. That's important when we go to Clustal and the tree. Clustal does this tree. And Clustal, in fact, does different trees, tries out different trees and sees whether they lead to different solutions. The beauty of Clustal is, at every single point here, for every single pair comparison, Clustal can decide, according to the similarity between whatever is compared at this level here, AB together with C, whatever, they can use a different substitution matrix. So they can tailor the alignment to every comparison at hand. That's one of the reasons why Clustal is very successful. In fact, most likely Clustal is the most quoted paper, most quoted method on Earth, any discipline. The people behind Clustal, the original one was Des Higgins, Toby Gibson, and Julie Thompson. Clustal is a sequence profile method. Another one here is Maxom. Maxom, the philosophy of Maxom is slightly different, although I sort of implied that before. It starts from the same, you see these things look the same as before. The major difference for Maxom and Clustal or PsyBlast is a protein-specific view, as opposed to a family-centric view. What I mean is, when I start from this sequence in the view of Maxom, this sequence is a real sequence. Everything that is aligned to it may have an insertion, may have a deletion, so we may delete some residues, but not in my first sequence. The first sequence is what I want to compare it to. That's my protein that I want to study, and I want to know everything about it. I don't care about the family, I only care about my protein. And everything here will be enslaved to the fact that this has to stay intact as a letter string. Here in Clustal, that's not the case at all. Clustal tries to find the alignment that is optimal for the entire family. In this alignment that is optimal for the entire family, it might be that I have to insert things in my particular protein, or I may have to remove some of my residues that might produce the best alignment for the family. Again, that is the view that I want to have if I care for evolution, if I care for average features of that family. And that gets us to the next one, which is the hidden Markov models here. They are slow, they need pre-processing, but they are getting increasingly powerful. The daylight zone, and I get back to hidden Markov models in a moment. In the daylight zone, I can intrude with sequence-sequence pairwise comparisons. The twilight zone I can enter into with sequence profile comparisons. Now, the way I build up these profiles again is I take sequence, get the results, build a profile, refine that, do profile against sequence, update the profile, and I iterate over that. Another way of doing this, same thing, is by math, by hidden Markov models. The simplest hidden Markov model here is this one. You have a sequence of events, 1 to n, a transition probability, and a probability of observation. Now, 1, 2, and are the states of the system. They are hidden. That's why it's called a hidden Markov model. The observed symbols here is what comes out with a certain transition. From the original state, you compile with a certain probability in output. That's the observable. Okay? So in this particular case here, different states could lead to the same ABA. And by the way, this is a fantastic introduction from Sean Eddy, published in Bioinformatics, for hidden Markov models. I believe this is still the best thing to start with when you want to understand how hidden Markov models can help in alignments. These slides come from Marco Punter, and Marco likes to play football. So this is 2013-14, the Bundesliga, the club is Augsburg. And for all the games that Augsburg played, home here, the score. Away, attendance. Now, you may want to predict simply the outcome. So you lose, you win, or you have a draw. That may be what you want to predict. That's then the output of your state. And that would then be a probabilistic model here, win, draw, lose. You combine all the model. In this particular case here, you would compile somehow this output according to the input. And one input feature here that you could possibly compile is whether you're at home or not. Whether you play at home or somewhere else. Maybe that is the simplest model by which you can predict with some probability W, D, or L. Win, draw, or lose. In this particular simplest case, home, away are your input variables. You build up this entire Markov model from the data that you have. Okay? And the formalism now is you have an emission probability here, you have a transition probability, those are the states and those are the symbols. That's again in the case before the 1 and the A. Okay? And you simulate essentially an observation. Now here's another example. Traffic light. And we all see the state of that system. What's the difference between the two? Between the traffic light and, for instance, the prediction of the weather. I'm going to show you another example, yeah?
The transition in the states is one way.
It's one way.
If it is red, it can only go to yellow and then to green.
It's very clear. Yes, it's deterministic. You know exactly how it will proceed, right? While, for instance, the weather example, we could predict the weather, sun, cloud, or rain from some simple parameters, and that's a purely probabilistic model for which you in fact have to compile the entire probability. Let's give you another example. The example of the dice. In fact, we have the occasionally dishonest casino, and again it comes from Sean Eddy. So there's a fair die and a loaded die. So the red one does not give you the equal probability of all 6 numbers. That's what loaded means. The fair one, so the probability of getting a 1 is the same as probability of getting a 6 for the blue one. Now, in the occasionally dishonest casino, what you will do is you will have a certain probability of going from the fair die to the loaded one. You will not always use 1, because immediately this would become apparent to anybody who sits there. By switching it once in a while at a certain probability, you hide the fact. The point, however, is, so here's the probability distribution. So in 75% of the cases, you will go from a fair to a fair. In 25, you will go to the loaded, which you will sample at 60% of the cases, and you go back at 40%. That's the sequence of the event that, in fact, the one who throws them decides. You can plot that as a probability distribution. That, in fact, the probability distribution now for the sequence of events, fair, loaded. That's the sequence. And you want to get at that sequence from the probability values that he have. And you simply can do that from the background probability of observing the numbers. Now, once you have this entire model decided, you can essentially simulate what is the most likely outcome of the sequence of numbers of this switch. And you can look at any given sequence of numbers and see how probable that particular sequence that you observe, where these two dies, is given the frequency of switch. Right? So it's a perfect way, the hidden Markov model, it's a perfect way to play for money. No, that's not what I mean. It's a perfect way to look at the probability distribution of observations, or to put probability distributions of observations into a formalism that is easy to compute. How do I do that for alignments? Well, in principle, the same thing. For every single, I start at some point, then I have an insertion, deletion, or a match state. Deletion, insertion, match state. Okay, that's when I look at two different pairs. So I want to align two sequences. The second one I ask, does it match? Should I insert? Should I delete? Okay, those are the probabilities. These probabilities I get from the family. So I begin this procedure with a family of proteins. The family of proteins are used to in fact decide at every single position what's the probability of deletion, insertion, match, and what particular match. What particular match then at one particular position means the probability of observing a P, S, or T at that point, or in the case of the consensus sequence, the two V's or the two LA's or something like that. Here is one particular solution, essentially the same that is in SAM, that was developed by Kevin Karplas. Again, we started from a single sequence, built a model. Now, in the first round, this is where previously we used, in the first round, we essentially used pairwise alignments, or in order to compare two strings, we used the comparison matrix, the BLOSUM matrix, the normal substitution matrix. So here what we use is a simple entropy, probability of observing. So it's minimal for a peak distribution, and maximal for a uniform distribution here, where all states are equal. You simply look at the microstates before any amino acid is aligned. So if that is my sequence that I want to line into, consider residue at the position I. Before any amino acid is aligned, we expect a particular amino acids to some prior background probability with an entropy HO. So that is essentially the same thing as the substitution matrix that I showed you before. That is where you can put the frequency of the entire database, the frequency of human, or the frequency of that particular protein family. All of these are different background models. Now, once I have a column aligned, I have an observation that is just the profile observation that I had before. The same thing here. So I, in fact, again here, the height of the letter is proportional to the occurrence of that letter. In this particular case here, the numbering is aligned with the residue running. So that's the first, second, and third residue, and so forth, for a protein that in this particular case is 37 residues long. And at every single position, again, like in a profile, you have a probability of observing a particular residue. Now, that particular position gives you the entropy and gives you the probability of getting a next sequence in there. Okay. One problem here is that when you align two proteins, you see conservation trivially, because there's only two, obviously they are not very diverse. When you align 200, conservation means much more. But it's not that easy to normalize that out. You could sort of normalize by the observation, but again, it's not trivial to do that. And that is one problem here. For that problem, you can imagine that you sort of have a slider that if you have few aligned or very little divergence, you essentially take the background frequency. And if you begin to see a signal with a lot of diversity, you move in the specific profile. So you go from one extreme to the other. And in between is not really clear how to do, in particular because this is getting, like any machine learning device, into overtraining issues. The more sequences you have that are very similar, the more the profile will be in that direction of those similar ones, and that may not really help you to get the entire family. So again, you start from a single sequence, you build a model, use the model to search for additional, that's the iteration step in PsyBlast. You re-estimate the alignment, that again is all part of the iteration step, and you go into a cycle, and you end, and the same is true for PsyBlast, if you no longer find anything that changes the profile. Or you end at the given point after 5 iterations, or some fixed number. Sam. Another program here that I have skipped is Hammer. That comes from Sean Eddy. I talked about his paper about understanding hidden Markov models. Essentially uses the same formalism. In detail, Sam and Hammer differ, importantly, but on the level of what I want you to understand, using hidden Markov model for alignments, they're essentially the same. T-coffee. This is much slower, requires pre-processing, is a genetic algorithm. The genetic algorithm begins, so the beauty of a genetic algorithm is you can throw in a mix of very different things. In this particular case, for instance, it begins with a library of alignments. And in this particular case, it begins with a library of local and global alignments. Previously, I told you that sometimes you don't really know, is 2 hits out of 2 better than 4 out of 7? So are local alignments, global alignments, what are better? The beauty of T-coffee here, of the genetic algorithm, is you can simply toss them all into your mixture, your admix, and then see what is better. So you begin with a bunch of alignments that have been, for instance, created by Clustal W. In fact, the author of T-coffee is was one of the co-authors of Clustal W at some point. And then you simply look at some genetic algorithm in which you look for the best combination of fragments that you align. So in this particular case here, where we have 4 sequences shown here, Garfield, the last fat cat, and that example is from Cedric. Garfield, the last fat cat, Garfield, the fast cat, Garfield, the very fast cat, the fat cat. Those are the 4 alignments here. And the genetic algorithm simply looks at different combinations of these strings that are proposed by the original methods and finds different solutions. And then it finds the path through the different solutions, and it combines these different solutions and comes up to what in the end is the best possible combination of all the alignment methods that you have. Now you immediately see that this is costly. Previously I said that dynamic programming is slow. Here I say let's just create a bunch of dynamic programming outputs and put them into the beginning of a genetic algorithm. Some of you may have worked with this genetic algorithm. It is a very slow algorithm afterwards. So you begin with something that is 10 times slower than anything I described so far, and you make it even slower. But the great thing is you can use additional information. You can use information such as local, multiple, global. You can use information from structures, and you can use information from specialists. Those are little tags that come in your alignments, and then that say simply put more of that in there. And then you hope that the grinder, the genetic algorithm, finds the best path through it. And that if you put in the wrong information from structure, the expert, or global, it will just not take that again. It will just go over incorrect information, and at the end of the grinding, what you will have, the coffee is tasty, hence, in this particular analogy here, this is really the right alignment. And there is no other tool that I'm aware of that can in fact do that. There's a unique combination at a unique price. It takes a long time. There are other methods today available that somehow find paths through these different things. Most people today, most biologists use Clustal. Most people use PsyBlast, I would argue. In most publication, Clustal is used simply because it has a nice interface, I would argue. It's a very reliable method that produces a plot that most experimentalists can put into their paper. As simple as that. PsyBlast, any drug development today starts with somebody running many PsyBlast searches. Hammer, databases, PFAM. Nobody will do anything in biology today without having looked at some databases of families. The PFAM database is one example, and that uses Hammer to produce its family. So there are 10,000s and there are thousands of times used every day. This is more a concept that essentially no longer really exists. T-Coffee is a tool for specialists who have computers, so for computational biologists, or people with some understanding of computers and some understanding of protein structure and protein function. It has immense possibilities because you simply, that genetic algorithm is the only way in which you can combine very different traits, very different types of information. Without, it's not like a Bayesian network where, or a device such as that. Just a sec, let me finish my statement. In which you have to have a probability assignment for decisions. The genetic algorithm just randomly combines, randomly connects, and then decides whether it's right or wrong. Carlo?
I'm curious, what's actually the difference in computational time between, I don't know.
So the question is, what is the difference in computational time? That today against 55 million is about 20 minutes on your laptop. That is depending a little bit on how large the alignment is. That's in the ballpark of day or days. That is a little bit faster, depending on, well, mostly Hammer is used essentially to align against an existing family. So we have 10,000 already, and then you run it against the existing family, and then it's fast. But if you have to redo that family, that is in a similar ballpark here. T-Coffee, we're talking a week or longer. And then typically after a week you realize that some of your parameters were not right. So T-Coffee is something that keeps you happy for a quarter of a year in order to get really the good thing. That's the ballpark, quarter of a year, quarter of an hour. But again, so you can do things with that one that you cannot do. In fact, T-Coffee uses all of these as input. That I believe CSS search is a dynamic programming such as Clustal with all the bells and whistles much faster. Okay? Okay. Sequence profile comparisons. We take a profile that is specific to that family and align sequences into that specific profile. That helps us to get into the twilight zone. If we want to go further, we have to do something more. And that's something more, obviously, sequence, profile. That's the next step. So from pairwise to multiple to sequence profile, and that is now this idea that instead of now having a sequence compared to that profile, you do compare two profiles. In principle, this is very simple. Intellectually, this is a straightforward generalization. Everything so far, sequence was trivial. Sequence profile, you just put a probability distribution for every instead of a letter. It's also trivial conceptually. And now we have two probability distributions. It's also trivial. The problem is that again, we blow up complexity. And complexity, sequence, I told you from the start, it is very simple, but you have alternative ways in which you can define optima. And these alternatives, you have alternative ways in which you say that there's a gap open, deletion, insertion state, and all of these kind of things. In hidden Markov model, we learn all these parameters. There's a lot of freedom. Now, for the profile, you somehow square this problem, manifold. And again, it's not that easy to fix all the parameters. Here's an example where the profile may not be exactly what you always want. In this made-up example here, you see that there's one particular point here, so that's one protein, the second protein, third, fourth, fifth, sixth, seventh, eighth. This is what you observe. I highlight here. And in what you observe, you see some pattern. So these three have the same in both positions. For some of you may know that this is a positively charged residue, negatively charged residue, so that may actually be two residues that bind, that form a plus-minus bond. And the same is actually true here. Here you have replaced that interaction by a hydrophobic interaction. It's still a favorable interaction. It's very different in terms of biophysics, but you see that both of them change because they're actually binding. And you replace this polar or this salt bridge, so the charged interaction, by some other type of interaction, but you still bind, right? Now, what you see here, in gray you don't see that the sequences underneath do change, but in fact there is a coordinated change. 3 R's, 3 K's, 2 W's. When you just compile the probability of a profile at every single position, you completely lose that information. Okay? When you come with the next one, you do not want to align necessarily. Let's look at the consensus probability. So the consensus probability in terms would be most likely the W here. In terms of information content, the W would be the outstanding alignment here. But this really may not be it. It depends on what you have here, what you want to align here. And that is not at all captured in the profile. So, takeaway on this one simply is you may not always want profile in alignment, but let's see what we can do. Here's a sheep hack to do a profile alignment, and that comes from Dariusz Przybylski. And essentially the idea is we want to go somewhere profile here, costs a lot of CPU, and again in terms of CPU time here we're talking profile sequence that is in fact in the ballpark of 15 minutes, and this is in the ballpark of days, more than 15 days. So we really blow it up here. Well, how can we do that? How could we shortcut that? And that is in fact now reusing the idea of consensus sequence. So you simply take the substitution matrix here, in this particular case is the position-specific profile, and at every single position here, I'm going, I can mark, so my protein runs here, I, S, P, I, E, T, W, V, P, and so forth. And at every single position, I look up the most likely residue at that position for my position-specific substitution matrix. So I build an alignment. In that alignment, I look up what residues are observed. I compile PSSM. In the PSSM, I look up the highest probability letter, and I use that as a sequence. That sequence that I construct here is an artificial sequence. So it's much wilder. In some cases, in fact, it may be the original sequence, and the ones with the red stars here, it was a very different one. It's much wilder than what I showed before, because none of these residues, or in the high probability, none of them may be dominant residues observed in the alignment. An information content. But now you immediately see where I'm going, right? Now I have a consensus sequence. The consensus sequence is letters. I know how to align letters to profiles. These consensus sequences are the outcome of essentially distilling the information from profile. In comes a profile, out comes a sequence. Okay? And now it's clear. Instead of doing a sequence against a profile, I do a consensus against a profile. The beauty is I know how to handle that. In fact, I can do that with the same program that I showed you before, PsyBlast, in less than 15 minutes. 15 minutes is 5 iterations typically. So just one more iteration at the end in which I toss in the consensus. When I do that, let's not get into the detail, we do much better. So you can do much more accurate alignment without cost for the speed. You just do, instead of 15 minutes, 18 minutes or something in that ballpark. So it's a tiny addition. It requires you to do some database preparation. That's the downside. And in fact, this essentially is behind a method called HH-Blitz. It essentially uses that idea in a more generalized consensus sequence, when we no longer have really a consensus sequence, but we have a consensus that is made up of 219 letters. So these are states. And that's essentially probability. There's some more information what goes in here. But essentially, other than that, it's the same thing that I showed you before. That is, let's call it the poor man or the fast way to profile hidden Markov alignments. HH-Blitz runs and is at this point, so that's the publication from 2011 in Nature Methods, from the Switting group, who was in Munich, now is in Göttingen at the Max Planck Institute. So HH-Blitz undoubtedly is the most accurate method that we have today, and it comes at a speed that is not far away from a PsyBlast. So this is something that is affordable every day for every work. Teresa made this flashback, multiple alignments. I put the slide in here because some people like organization of tables like that. So we have the multiple alignment path here that builds profile, PSSM parameterization, logo representation, and builds multiple alignments. There's the HMM, hidden Markov model path, that leads the Baum-Wells optimization to set the parameters. And all of this is part of the HMM path. And in between, in fact, you use a combination of all of these devices, because the HMM starts typically with the PsyBlast alignment. So typically you put all of that together. That allows you now to get, well, not really into the midnight zone, but it allows you to go deep into the twilight zone. Let's repeat some of the relations in sequence and structure space. So first, got a sequence space in blue, structure space smaller, because the amount of sequences map to relatively few 3D shapes that we observe. Depending on how we count that, we could argue there are maybe 10,000, maybe 100,000 shapes that we know today. Well, we clearly know that this is 55 million. This is clearly more condensed. So in that direction, most sequences that are very different, so proteins that look very different sequence, may fall to a similar structure. And that is trivial, because that essentially means that blue is bigger than ochre or orange here. Similar sequences typically map to similar structures. Most sequences most likely don't map to any structure, meaning they do not exist, meaning that essentially they are not observed. So they will not fold, there will not be anything that survives in the cell. In some of these cases the carrier dies out, and some of these cases simply the protein doesn't work. And in the cases in which the protein doesn't work, typically the cell digests this aggregate of protein. Typically, similar structure implies similar function. Often, similar function implies similar structure. Similar sequence, similar structure. I showed you the relation. Now, what I didn't talk about, most proteins that are very similar in sequence also have similar function. But protein can have very similar sequences yet different function, and proteins can have very different functions and very different sequences too. Clearly, the map between structure and sequence is easier than function. And let's just now go into the midnight zone from the perspective of 3D structures. So I talked about the midnight zone. In the midnight zone I said I can no longer do the sequence alignment. Is there anything else in there, in that region, that I want to find? How can I find out? Well, I take all the proteins for which I know the 3-dimensional structure and essentially superpose all of those in 3D. No sequence comparison. I have to do it slightly different, so I take an unbiased subset and do one unbiased against all. And then I simply count for every pair for which I know the 3-dimensional structure is the same. What's the level of sequence identity implied by that structural superposition? Okay? And that's one way in which people have expected this result to look. 100% sequence identity, 75, 50, 25. And you have some distribution here. So up here is a high count, and then you go further down. Then random, so we have 20 amino acids, 100 divided 20 is 5, so random is roughly at 5 here. But this is wrong, absolutely wrong. This is fake data. In reality what we have is the observation is here. And there are different ways of counting here. The vast majority, when I do an all against all 3D comparison, the vast majority of all the proteins that have similar structure are here. They have about 10% sequence identity. Now, 10% sequence identity, look, this is what I observe. This is random, the red. And they are very, very close to each other. So most proteins that are related in structure have only maybe 3-4% or 4 residues on 100 more conserved than random. So you have to distinguish something. Now, that is a very, very small signal. On top, some of these here that are conserved may be conserved for other reasons, for functional reasons. They may be binding sites. There may be some other controlled sites. So that may not be even all of structural conservation. That is remarkable. What it implies most likely is that 3 or 4 billion years of evolution were enough to essentially try all the shapes we can, or to in fact try all the possibilities of getting at those shapes that we have realized. And it may imply that convergent to the same structure and divergent from the same structure are events that we can hardly distinguish because we are so close to random. So coming to this peak distribution from here, unrelated things, or from here, related things, may be equally probable. And that's a remarkable reality. That is the midnight zone. Those are the things that we can hardly find because there really is almost no signal. We can only identify them when we see structures. And sometimes profile-profile alignments allow me to get as deep down as that. And with that, I conclude today's profile comparisons. Thank you for your attention. Are there any questions? Thank you.